this, and then we are indeed in short of lot of programmers, to help us navigate this stream and domain forward. Well, it's pretty much said about me. I'm not going to go in too much details again, but I'm so glad to be a part of this. And then uh, today we'll cover on the concepts of what is clinical trial as whole itself, and if you are aspiring to be a programmer, that are programmer, where do you fit yourself in these clinical trials, and how actually programming is done in a clinical trials. So I'm not going to go very much detail in the coding and all, because we usually offer demos once everything goes well and people enroll in the batches. But this is just to give an overview of what happens in actual industry and why programming is as a part is very very crucial in delivering the molecules to pharmaceutical products.
इंट्रोडक्शन दिखाने यार ये तुम दूगो काहे पहने हो ए ई हाँ कहाँ दूगो है दूगो है
I
नहीं
would request people to be on mute.
दिखा रहे हो अरे ये नहीं थी बोलेगा
Yeah, thank you. Thank you so much. Okay, quickly let me share my screen. Uh, just a second. Okay. So can anyone confirm that you see my screen, please?
Yeah, it is visible.
Thank you so much. Right. So as I just said, let us first quickly get into what is a clinical trial, right? So let us understand very basics of it. I'm not going to go in very much detail because we are well aware what the clinical industry works and how the drug comes into the market, right? So whenever a company decides that we have identified a chemical substance, so I'm just saying that as a chemical substance because it's not still delivered or modified as a product, right? So let us assume it's a simple organic chemical substance, and then the test
Please
from
make
here
it
starts.
on full screen.
Could I request again to please be on mute if you're not speaking?
Sir, please make it the slide on full screen.
Okay, let me try that. Bear with me. Okay, looks good. Sorry, something messed up. Just a second. Bear with me, please.
Yeah, it is full screen now.
Okay, so you're able to see the text now, right?
Yes.
Fine. So I just said, let us dig into clinical trials and understand the nuances. So when I said a pharmaceutical company decides to, create a molecule or a drug, it starts with identifying chemical substance, and then that chemical substance falls into preclinical development. So when I say preclinical development, there are people who are PhDs in science, PhD in pharmaceutical sciences, translation medicine, or generally as a whole as a medical fraternity. They get on these things. They start synthesis, purification of this, and then comes the formulation of how and we can modify this chemical substance. And once they have formulated, they have decided that a chemical substance is supposed to be a tablet, or it may be a vial, or it may be a syrup. Then they go for something called preclinical assessments on animals. So as a part of this preclinical assessments, they conduct this test on rodents, on dogs, on monkeys, which share very close resemblance with humans. And then after 3 to 4 years of extensive preclinical development, when they figure out that this chemical substance is actually working in an animal, they file something called investigational new drug. So where do they file this? They file this application in the regulatory country they're supposed to release the drug. So let's say if someone wants to conduct a trial and release the drug in India, they would submit this information to something called CDSCO. CDSCO again is a drug controller, organization of India. So likewise, for US, United States, you have something called FDA. For Japan, you have something called PMDA. So these are centralized bodies which will oversee, and they will decide whether a molecule has to be come into the pharmaceutical market or not, right? So once this IND is filed, they go into something called phase 1. So in the phase 1, it's very simple. They collect samples or volunteers of 10 healthy human people who do not share any diseases. Then they test the drug in this 10 or probably 10 to 20 humans. And then once they figure out that this drug is safe, how do they determine the drug is safe? They do something called dose escalation. So in the morning, they give 10 mg and then 20 mg, 30 mg. They see how much dosage can be given to a healthy person, right? And the ones they figure out that, okay, these people do not have any concerns, like none of them have died or none of them have very serious adverse events or side effects, then they move on to something called phase 2. In phase 2, again, they conduct the same research, but this time they conduct on people who are actually having a disease. So let's say if they have identified a molecule to cure a fever, then this time they take 200 to 300 people who are actually facing fever, and then they start giving the, this medication to those 2 to 300 people. Here they look at how effective it is, how quickly their fever is dropping down, how safe it is again, right? And then once this turns out good, they go to something called phase 3. So phase 3 again, this time you'll have thousands of patients involved. The recent well-known example for this phase 3 trials would be Covaxin trials or the COVID vaccine trials that we all were part of, right? And then after phase 3, they figure out that, okay, this works better in large number of subjects or the clinical patients ideally. So then we'll go for something called patenting it, right? So then, I mean, the patent starts from phase 1, but still this time they file something called new drug application. That means they've already identified a drug and it's working fine in number of thousands of patients, and they want to test something beyond the thousands or lakhs of patients. So this is what we call as post-marketing surveillance phase or phase 4. So this happens in lakhs, crores of population in a country that they select, the site they select, and then they start administering this drug and they see how safe it is and how effective it is. And they also look at what cost we should, put when we are releasing this drug in this country. And there's a lot of pharmacoeconomics and, all the testing that goes behind this. And then when phase 4 is completed, then the reports are then again submitted to government bodies, and then the government body decides saying that this drug is safe, you can release in the market. So that is when you're going to get your drug in pharmacy. So this is a basic idea of how drug starts from an organic molecule to a compound or a pharmaceutical product, right? So why I have to tell this? At every phase, if you see, you have colored blocks in this slide, right? At every phase, you need someone to analyze and send the data to government bodies, right? So how does government know that this drug is working? You collect the data from patient, you test the data, you see, you record the how much value of fever gone, went high, low, in how many days the fever was there. This is all the data that we analyze, right? So who analyzes that? At every stage, there will be a programming team who analyzes data. They will prepare the reports and then send to government so that they can be able to file a regulatory approval for the drug. Okay, so this is a basic concept in the clinical trials. And now you see a programmer plays an important role, be it in clinical development, phase 1, phase 2, phase 3, or phase 4. At every phase, you need a team of programmers who work behind the data and file the reports and send it to the teams, right? So now, as a general process, what happens? So we said the phase 1 starts and it ends at phase 4. But what are the teams that work behind each of these phases, right? The first team that works is medical affairs team or site monitoring team. So these people are experienced professionals in marketing. They also have high degrees of pharmaceutical or medical sciences degrees. They go to universities or to hospitals. Why do they go to hospitals, universities? Basically to find the patients, right? So that's where we get the patients. That's the place where we get the data of those patients. They go to these hospitals or universities, they speak with doctors, they select a particular doctor who is very well known in that stream. So let's say if a company is creating created an oncology drug and they want to test phase 1, so they would ideally go to a oncologist, the famous hospital, and then they speak with those investigators, and then they sit in something called IRB meeting where they discuss that so and so company has developed this drug and what is the pros and cons of this drug, how much patients we can allot, how much patients can be visiting our site, is this really a good experiment to do. So that is what the discussion that goes there during the approval process. Once this team approves, then the patient recruitment starts. Once the patient recruitment starts, the data flows. The data is collected, it is stored, and it is flown to the company. So when I say there are two different companies here, the company which goes to doctors and the hospital itself will hold the data, right? So when I say company, it may be a sponsor or a pharmaceutical company. In ideal terms, in a clinical research process, the pharmaceutical companies are termed as sponsors because they are the ones who are funding this research, who have funded it to make it, to a business product or a pharmaceutical product, right? So once the data is collected, we'll go much deeper in how data is collected on that in a couple of slides. This is just to give an overview, while I'll give a bit more depth to each slides when I talk. Okay, so when the data enters, someone has to enter the data, someone has to store the data, and someone has to analyze the data, right? So this is again the programmer's job to do it in any clinical project, in any clinical company, right? So the purpose may or may not be clinical research. In this slide, we are talking about clinical research, but there are many aspects where programmer can also contribute. They can also contribute to marketing approvals. They can also contribute to analyzing research, where telling companies that this is the place where we did not do the research, we have to find this molecule. How do they do it? They conduct meta-analysis. They bring the data from different data sources and they drive insights. So this is the actual job of a programmer, right? When we talk specific about clinical research, what they do, they collect the data that gets recruited from hospitals. They use certain practices. They analyze this data and they send to a data steering committee, or we also called as medical writing or regulatory affairs committees within the organization. They prepare the reports out of it and then they submit to government bodies, right? So when I say data is collected, in the previous slide, we just saw that after approval, patient recruitment was happening and the data is kind of entered and validated, right? So what is this process? How this step happens from the recruitment to validation, and where programming is involved in here, right? So when you see this single slide, the team that manages the data is called clinical data management, right? And within this team also, programmers do work. There are two sets of programmers who work for data management and who also work for statistical analysis, right? So in the previous slides, we just saw that a medical affairs professional talks to the doctors at the site, right? Second, let me reshare.
Unmute God. Okay. I'm sorry. Could you reshare your screen?
Yeah, I'm just trying to do that while I
I'm
figure out
really
the
sorry.
options a bit. It's okay, totally fine. Let me check if I can reshare it again. Hope you can see it, right?
Yes, sir.
Okay, perfect. Thank you for that. So let us come back. We were at a point discussing that a medical affairs professional visits hospitals and try to convince doctor that our company drug is so and so and we want to do a research. But how does he convince them? What's the source of document? The important source of a document when a research starts is called a protocol. If if in this participants, if there are people who have come from specifically pharmacy, they would know that by the year end, by the end of your graduation, they usually do a small kind of a project. And if I'm right, then you have something called a protocol, and you tell what is your research and how it is done in bits and pieces, and then again you do it a final report and submit for a graduation qualification, right? The same exactly thing happens at a much bigger level here. So the first document when scientists discover a molecule, that would be protocol. So they will start writing the protocol saying that we have discovered so and so molecule, and this molecule helps patients to reduce the fever, and we want to do a research in so and so sites in North America, in India, in it may be Africa, anywhere, right? And then they say the method how they want to do. They want to conduct observational studies, or where they want to collect the live patient data from hospitals. It's called observational study. And if it is if they call it as retrospective, that means data that is already collected in hospital. So there are n number of designs. So they would draft this document saying that who is going to work on this study, who is the doctor, or who is the site, how many number of patients they need for the research. Everything is documented in this single, document called protocol, right? And this is the document where the medical affairs professionals take to the hospitals and convince doctors or the organizations to say start the research in your hospitals. So this is the document which talks about our drug, and this is a document which tells why our drug is important and what patients are we targeting, right? So once this protocol is determined, we saw something called patient recruitment. Now, how do we recruit patients? What is the aspect? How do you collect the data? You cannot simply collect the data over a recorder or through an Excel, right? There has to be a way. If any of us, had a chance to visit hospital, maybe we were sick or accompanying someone who was sick, what do we usually do? We go to a doctor and we fill something called case report form. If you recollect any of your hospital memories, or maybe working there, visiting along with your, friends and foes, you might have seen that the moment we enter into a hospital, they've roll out a form where we start filling our name, our date of birth, our gender, our smoking habits, yes or no, and what is your complaint, something like that, right? So those are called case report forms. So those are unique for every subject. And then once we fill it, then we go for a doctor prescription, and then the doctor hands out a form and then says, okay, so this is what you have, this is the reason you have the fever, so take these tablets. So every sheet, every form of your information that comes out from a hospital is called case report form. And another example for case report form may also be your lab test. You go for blood test, and you go for urine analysis, you go for LFTs, you go for x-rays. Every test has a form where it will say how much normal ranges were and how much your range of blood was there, something like that. So every detail in the form is called a case report form. And this case report form, you cannot handle the data in paper, right? So imagine you are recruiting thousands of patients in India, and each patient has 50 pages of a book. So now if you want to do research, now multiply that 1000 into 50. So how much paperwork you have? We don't want to do research in the paperwork, right? So how do we store the data then? We convert those pages exactly into your forms. The best example would be your Facebook forms, or maybe any of the Microsoft forms that you might have filled, any survey forms that you do, right? So what happens when you fill a survey, you get a response saying so and so form is filled, but that response goes to the person who sent you the survey. It will store in a form of rows and columns, right? And he might have access in Excel, or he might be having access in any other formats, but you fill the data in a form, and that data goes into rows and columns and gets stored somewhere. So that somewhere is called a database. So that's where you see a term called DB. Then what is a design then? We understood what is DB, we understood what is CRF, and then what is the design aspect? Why design has to be here? So now when you're sending some Microsoft form or any survey form to someone, what is the ideology behind sending? Let's say if we are rolling out a survey after this webinar, what would be our intention of the survey? We will check your name, your email address, and we'll ask whether how was the webinar conducted. And then we'll also ask, are you interested to join the webinar? So you frame those questions, right? Exactly the same way, looking at the protocol, what research you are doing. Based on that research, these forms are designed. So now CRF is a paper form. That paper form gets converted into electric form or electronic form, and then it gets stored into some place. Imagine like a almara in your home, right? So when your data, imagine DB as an almara, so where you stock all your files. So now this electronic data goes and stores in your almara, but someone has to validate it. Why do you require validation? So do you think the data that gets entered at the site is always correct? There can be n number of people entering the data. It need not be always patient, right? So what if I have a data something like a male patient getting pregnant? Is it humanly possible? Is it legible data? It is not. Then we have to write some checks. We have to say that whenever a person enters a wrong data or highly impossible data, he should be having some error. Exactly like when you enter your Facebook credentials or Instagram and you give your password wrong, immediately it says your password is incorrect. So that is what we call as database validation or edit check programming. So this is where R programming and SAS programming also comes into part. Using programming, we design checks so that whenever a patient enters a wrong data that is expected, if he adds a date of birth of 2028, right? That's utterly a false data, right? So this is the scenario where programming comes into place, right? So once the data gets stored, once data gets entered, someone has to pull the data and then do analysis. What is analysis do you mean? We'll understand a bit more in the further slides. Till now, we understood how the research happens, how the patient comes, and how the data gets accumulated in your company. And then it's time to think beyond data collection. It's something called data analysis. So what is data analysis then? What is data analysis? Let us look at this simple slide. Let me see if I can. Okay, so I think you can able to see this simple flow chart, right? So if you look at this flow chart, it is very simple. With the protocol, we design the case report forms, and then the patient data gets collected in the case report form. That means imagine someone entering the data in your Microsoft Forms, and then the data gets converted into rows and columns. The first data that we get directly from the subject or the patient or from the hospital is called a raw data. Okay, so that means it's unclean data. It can have any number of things. It can have date of birth missing. It can have age missing. It can have gender missing. Anything it could be, right? And we have the responsibility to clean the data before we process that. Why do we process that? A simple example. Look at it here. So once we get the raw data sets in the clinical research process, since we have a huge responsibility over patients, the prime importance of clinical research is to handle the data without any deviations of the policies. What is a policy? Or I would say rather call it as a privacy. So I cannot collect patient names. I cannot collect patient phone numbers or address. So who says that? There is compliance in the research. So this is laid down by something called ICH, International Committee of Harmonization. Under ICH is a very central body under which every country bodies or every country drug regulatory authorities abide to. As per this ICH rules, you have to follow certain standards and practices when you collect the data, when you store the data, and when you present the data, right? So what are the standards then? The standards that are applicable in the clinical research, especially in the programming and data, is called CDISC standards. Right? So under CDISC standards, there are n number of standardization policies. But what exactly applies to our mode of work, especially if you become an R programmer or a SAS programmer? First thing is called C dash. So in the slide, if you see PRM, this is something that is dealing with the preclinical data. So usually programmers and all do not effectively work on a preclinical because most of the times biostatisticians or bioinformatics work at this space. Our role of work as an R programmer starts when the data is collected from the CRF and stored in the raw data. So when a person enters the data from CRF and gets into raw data, the standards that applied till this flowchart is C dash. Right? Once we have a raw data, we have to clean that data. right? So that standards is called SDTM. Again, what is SDTM? Standard data tabulation model. That means once you have the raw data from hospitals, you have to make sure that your data is clean. You have to make sure that you have subjects with correct date of birth, full date of birth, no age missing, right? And no gender missing, something like that, rules and regulations. And then we apply something called SDTM practices, and then we convert it into ADAM. What is ADAM? So if you look at this slide, if you look at this slide, if you see the data here, this is an ADAM data. Why? When I said SDTM, I meant standardizing the data into tables and rows, right? Rows and columns. When I say ADAM, it's called analysis data model. That means the data that comes into your system, you have to apply analysis. What is analysis of the study? So that means in my protocol, they say that I understand or I manipulate my data, analyze the data using ANCOVA principles or ANCOVA statistical methods. So I applied ANCOVA and I have all the statistics you see. Here I have derived N, I have given mean, median, minimum, maximum of the patient. So the basically if you see baseline is the first visit where the patient comes, right? So that means in my clinical study, there are 10 number of 10 patients and the mean of their values are 9.3. So that is something statistics I'm applying to this data. So this part of data conversion is called ADAM or analysis data model. So once this data set is ready, we applied ADAM, right? And then we call something called tables, listings, and figures because those are the end reports that you submit. So that's on screen. If you see, this is called a table. What is a table again? The exact replica of the data set that you have created. So I'm showing the same data in a form of a table here, right? I'm saying that my research study has 3 drugs, drug A, drug D, and F. And at the baseline, there are 10 patients. After the end of the treatment, there are still 10 patients. And then I've derived statistics for all the 10 patients. That means I'm summarizing the data, right? These are called tables. And then what are figures? The same data, if I plot something like this, so these are called plots or figures or graphs. You can name it anything. They are all same. So these are the end reports that go. The data sets and all, they don't go to the FDA. The end reports that goes to regulatory authorities are tables, listings. So we have seen tables and graphs. So what is a listing? If I generate the same data, something like a Word document, this is a data set, right? These are rows and columns. If I print the same data in a something called a Word document, the same data becomes my listing in simplest terms. Okay, so now where are we? We stand at the point of understanding the role of programming in clinical data management. And then we understood how the data comes from the protocol. And I mean, how the data comes from the CRFs, gets into raw data, and then what are the standards that we follow, right? And then how the data is converted. Okay, so I pause for 2 minutes here. And till here, if you have any questions, please feel free to unmute and ask me.
Guys, if you have any doubts, please raise your hands. I will unmute you. Raise your hands virtually. In the React option, you can find that one. Or you can type in also.
Yes, we also take questions in the chat. Okay, I'll give 1 more minute for people to respond. If I do not see a response, I'll continue the next slides.
Sir, could you please explain the third slide?
Okay. Do you mean this?
No, sir, the present one, the current one, which contain, yeah, this one. The CDIS standards in the clinical research process, that flow chart, sir.
Okay.
That is organize, plan, collect, and.
Yes. Okay. So in order to understand this, okay, so in the first slide, you saw the preclinical development, preclinical and phase 1 to phase 4 phases, right? If not, let me take you there. You see that? Preclinical, phase 1, phase 2, phase 3, and phase 4, right? Okay, so now as per the CDISC, I give this drug to animals, right? So from animals also I have to take the data. What data do you take from your animals? How much time drug was given? How much amount of drug was present in the blood after so and so time? And what were the side effects of the animal? Were the animal was shivering or fainting or died? And then how much lifespan the animal was there after giving the drug? What was the dosage you gave to animal, and what is that animal? You name the animals, right? So in order to collect that data in the form of a tables and in form of your Excel or tabular format, you have to apply some standards called PRM or SEND. These are the standards. So these standards programmers do not apply. These are automatically applied by the tools. Whenever they are collecting the data, the tool itself gets applied because they are compliant with that. As a programmer, as a real-time programmer, your CDISC standards typically starts from phase 1 to phase 4 because 99% of programmers are responsible for human clinical trials only. These are not human clinical trials, these are animal trials. So we are talking about clinical trials, clinical human trials. What is clinical trial? When a trial done in a human, you say that clinical trials, right? So this is preclinical trials. So now our programmer's work starts from phase 1 to phase 4. So if I have to collect the data of subjects from the phase 1, sorry. So how do I collect? I use a concept called C dash. What is a C dash? I am not going to go deep in it, but it basically understand in a very simple way that there are two companies. Okay, one company is working on oncology. Okay, now I'll take a simple drug. So let us assume there are two companies. Two companies are creating fever-related drug. That means if they give the drug to a patient with fever, that gets subsided, right? They do the research, they collect the data, and then when they are submitting to government bodies, one company says that our drug cures fever. Another company says that our drug cures pyrexia. What is pyrexia? People who are already familiar know that pyrexia is also a fever, is also a synonym for fever, because we have a common understanding. But we are talking about people who have done statistics. We are talking about people who have done auditors. They are not medical people. The person who reviews might not be a medical person in the government body, right? So now if every company starts sending their own terminology, then the person who reviews this data gets confused. So that's the reason the prime importance of CDISC standards is laid in place. That means no matter whatever you collect the data, you represent in the same way. Even if a company called Johnson & Johnson doing a research, or Novartis doing a research, or GSK doing a research, they have to collect the data in the same way, and they have to present the data in the same way, right? So that way of collecting data is laid down by C-dash principles, SDTM principle, and ADAM principles. Okay, I hope I answered your question.
Yes, sir. Sir, any software is also involved with this process?
Yes. So these are policies. These are not applications. These are like rules and regulations telling if you are collecting patient data, the column name should be use subject ID, but not patient number. Something like if you are collecting a patient, details in a Excel column, the column header should be use subject ID. What is use subject ID? Unique subject ID. So these are the principles which tell you how you should store your data, something like that. But there's no tool, right? We employ SAS and R to convert this. So that is the reason R and SAS are used to convert, organize, analyze the data following these principles. And in the further slides, I'll tell you how R can do it. Okay. So let's move on. So till now we have seen a lot of things about clinical research, getting data collected from different sites, and then what are actual challenges in data analysis, right? First, data collection itself is a challenge. So let's say your study is doing in 100 hospitals. Are you sure that every hospital out of that 100 is sending your data regularly? You have to monitor which hospital sent how much data, right? So for this, programming is involved. There is a team which called risk monitoring team, which will use this R and SAS or Python. They will identify what is the hospital, what is the country from where data is coming less than expected. Okay, and then again data cleaning. Okay, let us assume that 100 hospitals are sending you the data correctly, but how much clean is it? Are they sending complete data? Or are they sending just dates and months, not the year of the patient? Or maybe they are sending the patient details, but they're not sending the blood reports. So you also have to employ programmers in this space to see how clean is your data. So this is where your R programming and SAS programming are also useful. And then once you clean the data, so you also see the quality of the data. How much quality it is? Is it legible? Is it correct? When I say quality, I was referring to the old example, like a patient cannot put a future date in the data, right? He cannot be saying that my date of birth is 2028, April 2nd. So those data quality issues, who should check? It's the programmers team or the programming team using SAS, R, and Python. They check these data qualities, right? And then comes integration of data. So let's say your 100 hospitals sending your hundreds of data. How do you correlate? How do you put everything in a single place? How do you combine your data in a single sheet? Imagine there's a single Excel and every person is sending one column data. So how do you mix all that hundreds of columns and put in a single sheet? That is called integration in a simplest term. Who does this? Again, programming team using SAS and R and Python. Once you have your data integrated, it is clean, you don't have any quality issues, and then you have to analyze the data also, no? Because without analysis, what are you doing? What are you trying to prove? When you're doing a research, you frame a question saying that my drug will cure fever. How does it cure? What are the parameters that you look when you cure the fever? First thing, temperature should be down, right? But how much time you give to patient? If you give 3 times a day, is it temperature decreasing or increasing? So that statistical analysis who should do? There is a programming team again involved here who does the statistical analysis. And then once they do the analysis, they have to also interpret. Simply doing analysis doesn't work. They should tell whether it is actually curing fever or not. So that interpretation also the programmers work. So basically on the slide, you see a different aspects of data analysis. These all analysis aspects are done by programmers in real-time pharmaceutical industry. And how do they interpret results? Exactly like the previous slide. They interpret using these figures. They interpret using the tables. They interpret using creating a data, something like this. And these are the outputs that goes to FDA or any government bodies. They review this thing and say that, okay, you said your drug going to cure a fever. I see your data. It has really cured 10 people so and so in so and so time. Then we approve. You go ahead and market it. So this is the communication that happens between a sponsor pharma company as well as regulatory bodies, right? Now, we talk so much about the data, we talk so much about the clinical process, but what's the role of R? We are here in the webinar today to understand how R contributes this to programming, right? So far we haven't covered it. So let's see how R created, and in the further slides, let's see how R contributes to it, right? So the history of clinical trials and is way very before the Second World War or it. During the time, what used to happen is, you know, we all know that famous Nazi examples where people were tested with lot of superhuman drugs, something like giving poisonous gases, and assuming that the person will have extra capabilities. This is actually been done during German Second World War or before Second World War in Germany. That people used to die like animals. There were no regulations and rules how a research should be done on people, right? So clinical research is a way dated very back, right? But the programming in analyzing this thing came in early, probably I should say 60s in where SAS came, and before that there were a couple of other languages. But coming to R, so this R is a programming language and it is open source software. So what do you mean by programming language and open source? A software is a system. You ask question to a software, it gives response. So if I give the data and say calculate the mean, you have to communicate to the software, right? So you have to have your language. What is that language? You cannot communicate in English, right? So your software has to communicate you back and it provide the results. So that is what we term it as programming language. And if it is a SAS, we call SAS software. If it is an R, we call it R software. But what is this open source software? open source is something who nobody owns this. So SAS is owned by SAS Institute, but R is not owned by anyone. It's a free for everyone. Couple of people in university, they worked on some programming language that were already present, something like S, Pascal, COBOL, that were already present before 1990s. They have used those languages, developed a new language, right? The inspiration from this was taken from S. S was an earlier language where from S only R originated, and it got released in 1995. So that's where your first R started with a bunch of people called Ross and Robert in New Zealand and the University of Auckland, right? So this was merely used for small projects within that university and all. Later it evolved so much. The two people were joined by many people, and then many people were joined by thousands, hundreds, and today we have crores of users who actively use R. And when I say open source, these users also contribute their programming tips to the community. So if I have done an Excel, if I have done a data manipulation in 1 minute, someone might have done in 60 seconds. How did he do it? That piece of code he puts in here, saying that I have done it in a better way. So using this community where crores of people actively contribute their experiences, this actually has a huge impact. So that means a company need not employ 10 people to think on application for 3-4 months and build it, because someone in open source might already have built it in another company, and they can take that without any patented thing, without any problem, they can use it. So this is what we call as open source true meaning. Okay, and this is widely popular. This R program is been widely popular in statisticians, data science, and research who predominantly work in agriculture research and maybe medical research and pharmaceutical researches. So you can also ask me a question, sir. We have seen that clinical trial analysis was earlier done by SAS. Is it still done? And why now R? Why not only SAS? So if I have to answer this question in a very simple way, right? SAS is a closed source software. That means SAS software is proprietary to someone else. If I have to use that software, I have to pay to that company to use it. But if I have to use R in a company, I don't have to pay to anyone. It's free. So that's the cost effectiveness of this R is very, very crucial in these days. Pharma companies invest billions of dollars in developing a drug. The only thing that they look at is profits, obviously, because it's a business. And when you're cutting down your investment in something like employing open source softwares where you don't want to put money to buy software or to work in software, it's a huge profit for them, right? So that's where current pharma companies slowly started moving from SAS more towards R. I would not say that SAS would completely go or R will only be there. No, both are used in different places because each of these softwares have their own plus and minus, to be very honest. Okay. So when we talk about R programming, how the R program is fundamentally built, I said something about open source software, right? So how it is fundamentally called open source. I'll give a simple example of 10 different chocolates in a box. Assume this a simple example. So I call the box as package. So I'm talking about package in R. So now what is a package again? So imagine you have 10 different chocolates and you have put 10 different chocolates in a box and you have wrapped it. So now your 10 different chocolates are inside a box, and that box I term it as package. So in the programming way, I have to tell you, package is nothing but a bits and pieces of codes used for multiple purposes. They are organized together, right? And like that, you have multiple packages developed by multiple people. So let's say, for example, I'll take myself. So today I worked on something and then I figured out that I could bring the data from Excel. I could bring the data from a Word format or a PDF and convert it into a into a beautiful graph. I could do it very easily. So what I'll do, I'll showcase this how I did, right? I could showcase this how I did using a code. I'll say I used this so and so code and bought different files from PDF and Excel and all and created this beautiful figure, right? And I document it in a proper way. And I give the code example also how I worked. And then I'll call it as a one function, one purpose, one function. So like that, I have created 10 functions. So now these 10 functions fall into a box. It becomes a package. So single package has multiple functions for different approaches of work. Okay, and then where all these packages are stored. If I'm creating a package, if I've developed some code and I want other people also to use, where do I send? Whom do I send? I'll send to something called repository. What is a repository? Imagine like a bank. So there are 10 different accounts. Every account is a package. Everyone wants to, store their money to a bank, right? So everyone has a different accounts. So everyone has their own packages. Now they want to store their packages. They would ideally go to a place called repository and then store their 10 different packages there. So that's a centralized place where people develop the code, they convert packages. In very simple way, package is like a zip file. Inside a zip file, you have 10 codes. And that zip files gets into a bigger place that's called repository. Okay, so what are the famous repositories in R? There's one repository called CRAN. It's called Comprehensive R Archive Network. So it is a place where programmers around the world, they create their own codes. They think that the code is working fine. It is helping some way to other person or to a business purpose. They put that code as a package and send it to CRAN. And then the people in the CRAN, there will be, something like reviewers, right? So they evaluate the package and then they list that package in CRAN. So that means I can go to a website of CRAN, download that package and use for myself. That might be developed by any person in any world. Okay, and then the similar repository called bioconductor repository. So this repository is very specific for people working in bioinformatics. So in CRAN, you'll get packages or codes for all pharmaceutical, medical, banking, aviation. It could be any use. But for bioconductor, you only have bioinformatics related codes only or functions only. And then we already know something called GitHub. It's very famous. So GitHub is also a place where everyone has their own folders and then they store their folders into, then they store their codes into folders. Imagine like Facebook. Imagine like you have a Facebook like thing and you store your images in the Facebook. Everyone with your name can search your image, right? The same example is a GitHub. You develop code, you develop in your own GitHub account. If you give your account name to me, I can come to your account, take that code and work. I don't have to think about should I develop the code or not? Because it's already developed, it's already evaluated. I can simply use it for my purpose if it fits. So this is a concept of R programming. R programming is a mixture of packages that are taken from different repositories. Okay, and then when I say, how does R looks when you start working on it? Right, so R previously used to have a desktop version. So currently also you have a desktop version. That's another story, but otherwise you can install R in any computing environment. You can install in Mac, you can install in Windows, Linux, any computing environment, and you can use both desktop or well as your cloud-based. What is a cloud-based? Again, I'll take an example of Facebook. You have a Facebook account, you have a username, password, login, your Facebook opens. So that means you don't have to have a dedicated phone. Do you think your Facebook account opens only in your phone? You can open your Facebook account in any phones, right? In any desktop, in any tabs, in any of the computers, right? The same way, R also offers a cloud-based service. It's called R Studio. That means you can open R in any computing environments. You can also open in phone also, right? But what do you need to have? You need to have an account, R username and password. So when you open your account and you log into your R, so this is my dedicated R space. I see that this is divided into 4 screens. So this is the area where I write the code. And this is the area where the written code gets compiled. That means here I write the language, I communicate, and then this is the place where R tries to understand what I've written here in the white space. And then once it understands, it gives me the data on the right side of it here. So you see ADAE, ADAE, CBind. These are all the data that I've created using some code. Unfortunately, there is no code here, but this is just an example. After I write a code, this console understands what I've written here, and then it creates me the data that I want, and then I can store the data in my computer using this place. So this is called a script area. This is called a console terminal area, and this is called an environment area, and this is called files and folders areas or a version areas. This is how predominantly your R looks, and this is same in anyone's computer if you are opening a RStudio. Okay, and then So why R is used for clinical trial analytics then? We have kind of almost summarized everything, but let's see why R is being very much used recently. R has a very good statistical tools. So do you mean that SAS and other languages do not have? They also have it. But why R is different? R helps you in something with very good, image quality or very good rendering quality of your outputs. Imagine you're watching your movie in 4K versus 1080p. So what is 4K? It's Ultra HD, right? So R is capable of creating those Ultra HD or very clear images of statistical things, right? And the tools also, it's very user friendly. Then when you compare with other programming languages, right? And then there is something called improved data presentations. So there are some packages. What is a package again? I said package is a combination of code, right? So there's one package called ggplot2. What is this package ggplot2 does? So this ggplot2, you can create your bar diagrams, histograms, your graphical things like something like, a pie diagram, pie charts and all using this ggplot2 package. You just have to give your data and you have to tell on what parameters you want to create your pie chart, right? And then it creates beautifully. So it has improvised data presentation things. And then there are few packages called Markdown. What is Markdown? Markdown is something again is a code based thing, is a package which will help you to include your images, your code, everything into a nice PDF kind of a document. And you can share that document to anyone else like me or you or anyone else who are interested to learn. So it's very easy in sharing, or communicating the data with one another within an organization or a team. And then the level of customization that R provides you. Literally, you can create anything in R. Using programming, you can create an image of Sachin Tendulkar in R. Believe me. So I'm just giving an exaggerated, thing. But otherwise, yes, you can you can create that using code. But what I meant to say here is, there's no limit to your creativity in R. You can kind of create anything using code in R. You can create web-based applications. You can create games also to a certain extent, and you can create statistical applications where a single drag and drop you get very beautiful bar diagrams, histograms and all, right? And you can create something like any tools that takes the data in, sends the data to different formats and all that stuff. You can do a number of things. And then it is very easy to integrate with other databases. So that means, what is integration with database? So in the previous example we heard what is a database, right? So database is typically a place where entire your data gets collected. Now imagine in a company there are 10 databases, right? There are 10 databases. So using one programming language, you can fetch the data from multiple areas very effectively, very quickly. Okay, and then there is something called predictive modeling. What is predictive modeling? I'll give a simple example. In clinical trials, there was one trial, that was done on Alzheimer's. Alzheimer is a memory, loss disease where people lose their memory on after certain years of their life. Okay, so what happens? This thing is, there is one gene in our brain which will make us forget things after we reach 60 or 70. What is that gene? So what is predictive modeling and how it is applied here? Using programming, you can actually predict using blood reports of a patient. You take whether the gene is present in the person or not. Then you can calculate approximately how many years that is going to get this Alzheimer disease, or even if he gets the disease or not. That means you are predicting the disease before it is happening. That also can be possible using R. And it has large number of machine learning, data science capabilities to give you that analysis. Usually in oncology studies, what happens? We give a drug to a patient, we take the data of the patient, and then we call something called survival analysis. So that means we see if we give this drug to this patient every month, how many years he is going to live. I'm not talking about 1 or 2 years. I'm predicting if I give my company drug to this patient, taking his data, I'll calculate something to analyze after how many years he might die, or after how many years, his quality of life may be depreciated. So this type of capabilities can be easily solved in R, right? Again, cost effectiveness. We have also seen, since this is open source, companies do not invest much on this R. It's always free, right? And then community support. So when you get a doubt, you have crores of people already working on that. Anyone can answer the simple question. And it is very kind of hard when you go to closed source softwares like SAS. You have only dedicated people, SAS community where only structured questions are answered. But in R, you can find n number of things on Reddit or maybe in any other channel where people are already discussing your question. So that kind of community support you can get. And then regulatory compliance. So earlier, when I say earlier, I'll date back to 8 years back. There was kind of assumption that FDA only accepts SAS, nothing else, right? But slowly the FDA, when I mean FDA, it's a Federal Drug Administration. It's a largest body to authorize drugs in USA. So this regulatory authority kind of looked into the scenario and told that we need data sets, we need reports. We are not concerned about what software you use, right? So when FDA is not concerned about the software we use, then and there, the open source softwares came into picture. People started saying, okay, why have to pay money for SaaS software? Why can't I bring R in my company and do the projects in R so that my cost reduces and I can submit the same reports? Because SaaS and R eventually generate the same reports. Using SaaS also we generate TFLs. Using R also we can generate TFLs, right? But there are few challenges that we'll discuss in coming slides. But otherwise, so that is where you have regulatory compliance now. And you have companies like Novo Nordisk, GSK, Pfizer, or Roche. They have already submitted clinical trial projects in R and they have approved their drugs in using R programming language. So the future is bright for people who are coming into R programming. And also the future is also very, very bright for people who are already in SAS and they're started working on R. I myself started working in R. I come from a SAS programming background. I have to learn R to sustain in this industry and to move forward. Right? And then the time efficiency. When you run programs, when you set up your studies or when you set up your data manipulation things, the amount of time that takes in R is very less. It can handle a large number of data. It can handle a huge number of people working at the same time on your data and all. So these are the top reasons why R is very used in clinical trials. Okay, and then welcome to R. Till now, we have only seen why R is important. Now we'll see what R can give us. So within R, we have understood what is a package, right? So when you install R, you don't have to go to CRAN, you don't have to go to Bioconductor, you don't have to go to GitHub. Why? Basically, you have the inbuilt packages. When you install R, you have inbuilt games in it. Imagine like that. So that inbuilt game is called R base package. It can do all the work basically. But the only thing is you have to write n number of lines to get an answer. Then sir, why do we use packages then? What is this tidyverse and farmverse then? When we already have something in R already, why are you pointing to arrows towards something called tidyverse and farmverse? I'll give a simple example. So let's say a person just installed R and then he wanted to import an Excel file from his computer. He installed R, he started writing the code, and he literally wrote 10 lines of code to get the file into his system. Right? And then again, second day, again the 10 lines of code, get the file in system. But later he realized that some clever person already created a small package where using that package, he has only to give the name of the file. That's it. No coding. The 10 lines of code gone. He has to give only the file that he wants to input. Rest that package will take care. So now tell me, in a realistic way, do you write 10 lines of pages every time? Or you'll go to CRAN or Bioconductor and get a package where you can simply give a file name and the work is done. Right? Ideally, as a human tendency, we look at the things which are very simple and we are faster to do it, right? So that is where these packages come into place. So I term this package as base package when you have to write a lot of lines of codes, and then the same work is already done by someone else, creating a simple function or a simple name. You just have to use that name, give the file name, and then your code automatically gets written in the background. So those are called advanced R packages. So in the advanced R packages, the important package is called tidyverse. So in tidyverse, imagine it's a box, right? Imagine a chocolate box. Tidyverse is a single box. Inside the tidyverse, you see a lot of chocolates here, which talk about Diplar, Tibble, Readr, Stringr, ggplot, Forcats, Purr, Lubridate. What are each of these? These are the chocolates inside that package. So package is tidyverse. These are all again individual package, but still these are compiled into a single package. So why Diplar is used? Diplar is used to get your data into system, convert your rows into columns, columns into rows. And then why ggplot is used? Is used to generate statistics or graphs. And why Tibble is used? Tibble is used to categorize the data into a data, properly rows and columns. And then what is tidyr? Tidyr is used to clean the data. Like let's say if any patient date is having missing month, how do you handle that? That can be done using tidyr. And then what is Readr? Readr is a R package which helps you to fetch the files from outer environment, right? And then Stringr, what is Stringr? Stringr is something like if you want to find a particular patient in a simple example. Imagine there are 10 lakhs of patients in a file and you want to find a patient who enrolled in US so-and-so hospital. You have to apply a filter, right? So that filter can be done using a Stringr. And then what is Forcats? Lubridate is something that deals with the dates and all. So each of these colored chocolates, so you see, which I term as packages, they have unique function in R. And the codes are already written in the background. You have to just take the name and pass the data inside it. Okay, whereas if you want to employ your R base, then you have to write your code also. Okay, and then we have spoken about tidyverse, right? So tidyverse is a collection of R packages specially designed for data science, right? So when I say data science, it can be used for data cleaning, data analysis, quality checking, statistical analysis, interpreting everything. And then what about SDTM, ADAM, TFL? As a clinical R programmer, we have to have packages that are useful in clinical programming, right? We don't want something to use in data science. Then how and what we should do it? This question has been asked 8 years before, and companies like Roche, Johnson & Johnson, Bayer, GSK, and Novo Nordisk, Eli Lilly, these companies came together. So when I say came together, how do they come together? Do they sit on a coffee table and discuss? No. A programmer from their team who leads particular initiatives, they all get together in meetings. And then they say that we have to come up something with inbuilt packages that every one of us can collaboratively work and that should help us in clinical programming. So with that idea, the formation of Pharmaverse came. So what is Pharmaverse? Pharmaverse is a connected network of companies where people from different companies, high profile programmers, they come to a place and then they work on common problems and create solutions there so that everyone can use. I am myself a part of one of those beautiful Pharmaverse packages in R. So we look into something called submissions to regulatory authorities. If I have to submit my file, what are the rules and regulations? Is there any code that can help me to properly indent, create a table of contents, something like that? So we, as a programmers of different industries, come together and solve the questions. So under Pharmaverse, already there are a lot of packages that got developed under SDTM, ADAM, and TFL. I have listed it. So when I say SDTM, why we have used SDTM again? To standardize the data that is coming from hospitals, which was raw data, right? So when the raw data comes, you have to clean the data, you have to check the data. So now the SDTM checks package are pieces of code that is present in this package will help you to do that. And there is a lot of packages called Data Qatar, Oak, and ANCA. I'm not going to go deep because you'll get lost in all this information. These are simple sample packages or all inbuilt codes that you see available already for your SDTM, right? And for ADAM, similarly, once your SDTM is done, the same data gets converted into ADAM. So in the ADAM, if you see, there is one beautiful package called Admiral. So this is what Roche has developed, and SDTM Oak is also developed by Roche company, right? And then you have very specific therapeutic area related ADAMs. Like say your clinical trial is an oncology trial. That means your drug testing is in cancer drug. Then your data has to be a proper way. What rules and regulations? You can do that using Admiral Onco. or your clinical trial is retina or eye related trial, you can use a package or codes that are present already in Admiral Opta. And your trial is COVID vaccine, then you can use Admiral vaccine package. So it has all inbuilt functions in it. You can generate reports, you can convert the dates, you can store the data, you can analyze, everything can be done using this. Right? And similarly, you have Admiral Pediatrics and Admiral Metabolism related. So these packages are actively being contributed. Like many packages will come in near future. People are continuously working in it. You should see their contributors are 84 people or 22 people. You see this? So these are stars that they get rated. This is like IMDb rating in movies, right? So based on the ratings, these get listed on CRAN. You see the CRAN here, and you see the symbol. This is GitHub. So that means these packages are available in CRAN and also in the GitHub. So there's separate area for Admiral in GitHub. You can access that area. You can download this package. Okay, and then again for TLGs, what are TLGs again? TLGs are the end report that we are going to submit to government bodies, right? So for TLGs, do we have any other things, any other packages? Yes, you have something called R tables. So which will take your data, create a report. I have showed you report in the 4th or 5th slider, right? So those types of tables can we generate in using R tables. And there's a package called Chevron. So this Chevron already has codes itself. That means if you want to show a blood report, you just have to give the data. That's it. The blood report is automatically generated using this Chevron package. There's standard templates, how and where you can generate it. Okay, and then there's something called Pharma RTF. What is Pharma RTF? So when you submit these reports to federal bodies or government bodies, how do you submit? You submit in XML files, you submit in Word documents or RTF files. It's also a Word document or a PDF. That means your data has to somehow convert into a RTF, PDF and Word, right? So this package will help you to convert the data. Imagine your data from a database is analyzed and getting converted to finally a PDF document. So that this Pharma RTF will do. And there are n number of things that that are included under tables. And then again, for listings, there's a package called R listings. We can efficiently take the data and generate into a Word document, telling what is the patient name and all the details that you want, right? And then in the recent example, I've plotted something about survival analysis. If you remember, I've said that we give a drug to a patient per month, and then we calculate how many years that patient goes to live. For that analysis, statistical analysis, you have a codes called ggSurfit, which will help you to calculate the survival analysis, especially if your drug is in oncology. And again, we also have our famous ggplot2, where you can plot your histograms, bar diagrams, pie charts, anything, line graphs, scatter plots. Okay, so having these flexibilities, it's very ideal for the pharma companies to switch slowly to R because they are already ready-made codes. No one has written, no one needs to write it everything again. They're already written. Anyone can use it without any regulations and rules. Right? So now let's see what are the organizations that are adopting R along with SAS. While you're talking about R learning, let us also understand what are the companies that already started working. So on the left side, you see the sponsors of pharma. So these are the main companies in the world that actively started working on R. So that means someone have already submitted project in R, someone have just started, someone has started 4 years back, 5 years back. It may be anything, but they have started using R. Okay. And what is this CRO? Imagine a company called Johnson & Johnson. So Johnson & Johnson is a company, it's a subsidiary of more than 200 companies. That means Johnson & Johnson will not only work on drugs, it will work on spectacles, it will work on medical devices, it will work on baby products, it will work on your Dettol, Listerines and all that, right? Daily use products. So do you think Johnson & Johnson can extremely focus on a single project for 10 years? It might be challenging for them because they work on different aspects, right? So what they do, they give that contract to another company. That another company is called contract research organizations. So that means tomorrow Johnson & Johnson company can give their oncology project to a company called Paraxil. So on behalf of Johnson & Johnson, Paraxil will work on that study. They will create everything, they'll create reports and then send it back to Johnson & Johnson. And this Johnson & Johnson submits to government. It's a contract. So that's why it's called contract research organizations. So now tell me, when a sponsor company is giving contract to another company, this company says, I'll use SAS. and the person who is spending money on this company says use R. Who do you think the argument wins? Is it the contract person or the person who is giving the job? It's obviously the person who is spending or giving the job to another company, right? So tomorrow, if R is being adopted in these companies, anyway, these people, even if they don't want to have, they compulsory used to, they have to use R because they'll lose projects. Because from Johnson & Johnson tomorrow they can say I can completely, I'll only work in R. I don't want to spend money on SAS. Then Paraxel should definitely listen to this guy and they should start investing only in R programming. So that's the danger here. So I'll not call it as a danger, I'll call it as a challenge and an opportunity to learn. So when your sponsor companies is moving towards open source packages, it's the responsibility of these people who are working with this also increases to move towards R. So that's why I say the future is bright for people who just learn R, or people who have already learning R, or people who learned SAS and coming to R. And then with this question, who can learn R? Again, reiterating, experienced programmers who already work and know SAS, Python, and are already statistical programmers in trials, clinical trial jobs. They can learn R easily compared to freshers. But it would be a nice continual learning for them. Anyway, their organizations will slowly move to R, right? And then freshers who find difficulty in searching SAS jobs can learn R, since in the current industry trend, there are only few people who know R in clinical trials. So the chances of getting higher after learning R is always high. So when you know R along with SAS, or you know only R, high likely they would like to take you into the teams, because this time is very, very crucial. Industry is slowly transitioning. When it is transitioning, you have to learn it. After transition or before transition doesn't matter. When it is slowly transition, this is a time where freshers will have adequate opportunities with high packages when they come into R programming. And I've stated the salary ranges. If you are just starting, your salary may be 5.5 to 6 lakhs LPA. With the number of years increase, it can even go to greater than 30 lakhs based on number of years. And personally, I know people who earn more than 80 lakhs or 90 lakhs having 15, 14 years of experience in programming. So there's no cap for how much heavy it would be the package, but the mindset always should be the continual learning. So tomorrow, if R goes and Python comes, you have to integrate yourself to learn. So I don't say that Python comes, Python or SAS. Even if you learn any of this language, you will easily learn the other language because they're all object oriented. They share a similar fundamental of understanding the problem, right? So imagine like this, like a maths. There's a math problem. If I give a problem to three people can solve with three different solutions, right? The only thing I want is the solution to be correct. I don't know what method you have used. So tomorrow there might be a challenge where you can do a project with whatever language you know. I want the result. So if the programming industry is going towards that, and this is a right stage where people should start exploring SAS, R, and Python in their day-to-day programming skills. And please note, for freshers who seek entry into the clinical trial programmer roles, do not worry, you do not need any programming prior experience to learn R. Likewise, if you are already proficient in SAS and Python, more than welcome. You learn in just 1 month. For freshers, it may take 30 days because they have to understand the fundamentals. Right? So do not hesitate or do not limit yourself saying that I'm coming from a pharmacy background, I'm coming from banking background, I don't know programming. No. We make sure that we start our batches, train you from very, nuanced level, like, very beginner level to an advanced programmer. So that's challenge is on us. When we come to course details, so we offer a 60-day course that would be 1 hour per day, and the timings would, we would decide based on morning and evening batch you prefer. The course details, the course also includes a CDISC project, which also includes, I'll teach you how to use SDTM, how to program SDTM, how to program ADAM, TFL using R. And every day notes will be sent through emails. And we have live coding classes. It's not a recording class or something like that. I would be coding lively, or the trainers would be coding lively, and you can ask questions then and there. We will help you out. And once the session is done, we record that session simultaneously and you have the access to the recorded session. So let's say you have just started the course and you have missed the second day, right? When you come back to third day, you don't have to worry saying that I missed yesterday's class. There will be recording. You can always go back to that recording, listen everything, come to us. And then all the materials, including software installation, assistance, notes, books, everything. I mean, like when I say books, I meant the supplemental, materials that you require for learning will be provided from our side. And then once you finish this course, we would offer you something called resume building. And we also talk about making you ready for interviews or job assistance in that regard, right? And if anyone are too specific about a demo again, we'll be happy to take that also a separate only programming demo. This was not a programming demo. This was a webinar indeed. So some of the people might think that why don't I see a code? So this was not to scare you in the first place. Okay. So if you insist that you want a programming demo before joining, so again, we can arrange it based on number of people, and then you can proceed with this. And then you have unique cross combinations that are available nowhere in the market right now. And if they are available very high prices or very less. So we offer clinical SaaS also, only alone clinical SaaS with CDISC. We offer clinical R with CDISC. And then we can also club both clinical SaaS and R with the CDISC project. So these are the available courses that we have. And we have a rough idea of starting a batch from 24th March. I mean, the next week. And we look at the entries, we look at the interest of the people. And we may have slight delay, 1 or 2 days before or after this date. But we would like to take your feedback. If you're interested, you can fill out the forms that we are going to share in this channel. You can come back to us. If you need further details, you can take my details from Kiran or Preeti here. You can speak to me, the trainer, myself, and then we can see how best we can help you in this regard. Okay, so with this, I end this webinar, and thank you so much for your time and patience joining here. And I leave this floor for questions. So I think Preeti, you can take it from here, and let's see if you have any questions on it.
Yeah, thank you so much. Thank you so much, Ashwath. And guys, do you have any questions to ask? Feel free to ask. And Ashwath, we already have a question here. Anirudh has asked, like, is this data cleaning related to clinical data management? Ashwath, you are in mute. You can unmute yourself.
Sorry, I was speaking. Okay, so that's a nice question actually. So if I have to reply you in a very basic way. So data cleaning is a responsibility for n number of teams. I would not say it's only for the programming team to do it. Okay. But how do you know the data is clean or not? Who looks at the data? The one who collects cannot look at the data. Like, in fact, I can look at one subject, 2 subjects, but if you have thousands of subjects, can you physically go to a place and look all 1,000 subjects records? No. So clinical data management, in fact, they're responsible data cleaning. But if they have to clean the data, first they have to identify what the issue is, right? So who gives the clinical data management the issues? It's the programming team. This programming team will work on the data. They'll see that so and so subject date is not entered. So and so subject age is not entered. And this is communicated to a clinical data management team. And then the clinical data management team will take the subject numbers and they'll check in which hospital these subjects are enrolled. And they send these queries to that hospital and that gets corrected and gets back. So the data cleaning as a whole, it might be related to clinical data management, but the predominant factor of raising issues or saying that the data is not clean lies in the programming teams. Okay, hope I answered your question. If you have anything to say, please go ahead. I'll clarify more.
Guys, meanwhile, I have shared feedback form on this session. I hope all of you have enjoyed this session.
Sir, I have a question.
Yes,
Yeah,
please.
please allow me a second. So fill up this feedback form, guys. Meanwhile, sir is taking question and answer session, right? So meanwhile, fill up this form, and your feedback is very valuable to us. Yes.
You have said about your course. So, sir, is there any charges of these course?
Yes, that will get back to you. So you have to reach out to us after this session to me or Preeti or Kiran based on the feedback that you share, or else we'll also reach back to you and we'll discuss further on that.
Arshi, please fill up the feedback form. I'll get back to you.
Yes.
Sure.
Okay. So Preeti, could you send the link again? I'm not sure.
I'm sharing again. I'm
Yes.
sharing again. Guys, please respond whether it is accessible or not. If it is not accessible, please inform me. Is there any other questions, guys? Any other questions? So how many of you are freshers? How many of you are freshers here? Okay, it is going to Anirudh personal window. I'm really sorry.
Maybe you have to say.
Yeah.
Yeah.
I'm really sorry, guys. I have already shared the link again.
So I'll leave this floor still for the questions, and do not hesitate to ask questions, because as a people we learn more than trying to learn alone. You feel free to ask any other question that might not be relevant to art programming. It could be any career oriented, anything.
Any career
Yeah.
relevant question also. If you want to ask, you can ask us.
Yes. So we can still take those questions. No worries.
If you are a fresher and if you want to start your career as art programming, you can ask us. Or if you want to ask about the salaries or the career growth in art programming industry, you can ask us. Any kind of questions. You can raise your hands also, or you can type in your chat also. We can take all questions. I hope all of you have filled up your feedback form, because it is very important for you, for us also, to take the feedback, to improve ourselves, to provide you more industry relevant webinars. So Rise Trainings always gives industry relevant webinars, you always know, right? So whatever the students demands or professionals demands, based on that one we conduct webinars. So why are you sending, when people are sending personal mails to me?
Maybe they should also do something called
Yeah,
meeting
they got
group chat.
disconnected, something like that.
Okay.
They are saying they are asking for feedback forms.
Okay.
Guys, I am again placing feedback form link here. I hope you filled up the feedback form. And if you have any questions, please fill up the feedback form. So if you can ask us, even your career relevant questions not related to the R programming, you can ask us. Okay. I hope all of you have filled up your feedback form. So I'll share the same feedback form in the group also. You can connect, you can fill up from the group also. And I also share, if anyone want to listen this session again. I can share the YouTube link also. So after this session, please ask us so we can share the YouTube link also. You can share with your friends and colleagues, right? So if we have no other questions, we can end the session here. And thanks for all of you who has joined here for this session, right? And we thank you so much, Ashwath.
Well, thanks so much, and I look forward for more inquiries. And then
Yeah.
please keep sharing this with all your friends and foes, or the people who might be interested in future references also to
Thank you,
work in
Puneet.
R programming. So yeah, with this, I think we can end the session. Hope we'll
Yeah.
meet again. Thank you so much, guys. Have a
Yeah,
great day.
bye all of you guys. Thank you so much for
Yeah.
attending today's session.
Thank you. Have a
Bye-bye.
great day.